Welcome to Chapter 9, everybody. We're in the last chapter of this series. This is all about how we control microbes. So in Chapter 5, we talked about how we can grow microbes in a lab and the way microbes grow in laboratories. In Chapter 6, we looked at their metabolic activities. So what type of metabolic activities do they have? And how do they produce the majority of their energy? And now Chapter 9 talks about how we can control their growth or kill them off if we don't want them, because not all microbes are beneficial. So definition-wise, the term sterilization means the destruction of all life. So if you sterilize an environment, you get rid of all life forms in that environment. All microorganisms, endospores, even the viruses, which are non-living but can still cause disease. Sterile objects can become contaminated, though, if they leave that sterile environment. So it's sterile so long as it's still in that whatever environment it was in. In our lab, we sterilize our inoculating loops. And within a few seconds, I don't want to say a few seconds because you have to cool it off to some extent. As soon as the loop becomes cool enough, bacteria in the air can actually attach to it, which is why we tend to be relatively rapid at the sterilization of the loop and then the grabbing of the inoculum to put on the new media. Sanitation, on the other hand, is just reducing the number of microbes or pathogenic microbes to a means considered decent by Food and Drug Administration or Association. I think it's Food and Drug Association. Sanitation also keeps microbes from growing out of control. So when you work in a restaurant, you tend to sanitize the equipment that you use. You don't sterilize the equipment. But if you're working in a hospital, you sterilize equipment prior to usage in surgery. So this chapter is all about how we control life, microbial life, but there's no one perfect method to control all life forms. There are multiple factors that are going to influence our choice. And so I like these cartoons. Here's a species reunion. I live in Pam. I live in John. And they're talking and getting together and chit-chatting. And down here, the bad news about your bacteria infection is that you are now part of their food chain. So that's kind of a creepy thing. But to control these microbes, we have to look at the number of microbes that are present. What type of microbes are they? Are they endospore formers? Are they viral? Are they eukaryotic? What is the condition, the environment that these microbes are growing in? If it's on skin or in the body, we're going to treat it differently than if it's, say, on a toilet seat. The risk that is involved in using certain chemicals. So you have to look at the risk of the chemical and outweigh that from the risk of the infection. So, what if the chemical is going to damage the body, then we might not want to use that unless it's the only chemical we know of that can kill off this pathogen, and the pathogen will kill the body, And then the composition. If we're dealing with skin versus a toilet seat, that's going to influence our decision of what type of chemical to use or what type of treatment. If it's a metal object versus a glass object, that's going to influence our decisions. So when we kill microbes, they do die similar to the way they grow, but they're not killed immediately. Just like the growth rate is exponential, death rate is also exponential. It's just slower than the growth rate, typically. Populations die in an exponential fashion. So typically death rate is exponential and slow. If we use chemicals or any other type of agent, that can actually kill them off a lot faster. And we use the decimal reduction time to determine the effectiveness of whatever agent we're looking at. And so what that is, it's the amount of time it takes to kill off 90% of the population. And so here we have a little chart that I made. This actually goes with a figure from your textbook, and it talks about reducing a population using the D time or the decimal reduction time. If you start out with 1 million bacteria, and the D time to kill 90% of the bacteria is 5 minutes, you have to get the bacteria down below 1. If you have 1 cell, then you're not going to kill off the bacteria. It would take us about 35 minutes. And here's how we know this. Oh, okay, I lied. It's not on here. Hold on. I'm going to take, I've never done this, so this is kind of exciting for me. I'm going to use a pen, and I'm going to just draw on here, okay? Okay. Oh, I lied. I'm not. I thought I was going to. Laser pointer. Oh, well, that doesn't really help, does it? So instead, what I'll do is I'll just circle that with my laser pointer, which is kind of cool that I have. I didn't realize I have that. And on Thursday, when we are discussing this, a lot of you may not understand how to do this. It's really easy. And so we'll go over this decimal reduction time, okay? So population composition. What type of microbes are we dealing with? Here we have Mycobacterium, which could be tuberculosis, or it could be Leprae, or it could be like what we have in our labs, Smegmatis, which doesn't cause disease. Over here we have Bacillus that produces endospores. Both of these are going to be harder to kill off than your basic like E. coli. And so different microbes have different sensitivities to antimicrobial agents. The environment, so the environment that the microbes are growing in. If they're growing in your body or on your skin, we're going to treat them differently than if they're growing in or on a toilet seat. My example is we wouldn't use bleach to clean our skin here, and we probably wouldn't use like iodine to clean the toilet. We also have to look at what type of materials are around the microbes. So there's a lot of dirt here. There's a lot of organic material here. That's going to inhibit some chemicals that we use. So we have to first clean the wound really effectively if we want the chemical, whatever we're going to use, to treat and kill off the pathogens. So one of the first mechanisms, and probably the most common mechanism of controlling microbial growth, is temperature. So we use heat, and there are two basic types of heats, dry and moist. And in using temperature, if we take and increase the temperature above, very high above the growth range, what they normally can grow in, then proteins are going to be denatured. Nucleic acids start to break down, and dehydration occurs. So we use, this is dry heat, this is what we do to sterilize our loops. Moist heat is one of the most common forms of heat, and it's a very effective means that we use in our lab. Here's another dry heat application, incineration. Incineration uses extreme dry heat to kill microbes or to kill whatever. And I don't even want to try and figure out what these are. It's a figure from your textbook. I'm showing it because it's showing incineration. It could be garbage. It could be that we're incinerating the garbage in a dump. I don't know. So dry heat is very effective, and we use it in the, and I should say you use it in the lab every week almost, whenever you're moving microbes from one media to another media or to a slide, you're using dry heat to sterilize your loops or your needles. Moist heat is a little bit different, and it's actually a little more effective than dry heat. You don't need the temperature to be as hot with moist heat, and it doesn't take as long to kill the microbes with moist heat. So one mechanism of moist heat microbial management is called pasteurization. So in this case, what we're doing is we're not sterilizing, we're just reducing the amount of microbes down, and we're getting rid of any pathogens and any spoilage microbes. There are two forms of pasteurization. The most common is high temperature, short time pasteurization, where we're going to heat some type of fluid up to a really high temperature, about 72 degrees Celsius, and we leave it there for a short period of time, and then we drop the temperature down very rapidly. So that, you heat it up to 72 degrees, and then you drop the temperature down very fast. And this kills any of those spoilage microbes, any potential pathogens. It doesn't get rid of everything, though. Then we have ultra-high temperature pasteurization. So in this one, we increase the temperature to close to 140 degrees Celsius. And we hold it there for just a few seconds. So like 3 to 5 seconds. And then we drop that temperature down to, that really cold temperature of 4 or 5 degrees Celsius. And what that does is it kills off microbes so effectively and gets rid of all those spoilage microbes that then we can package the material and store it on shelves. And so this is how we make or we produce milk that is shelf safe. In the US, not a lot of people purchase shelf safe milk. Sometimes you might if you're buying those little like milks or drinks for like a lunch. but most of the time people buy milk that is in the refrigerator section and has to stay cold. Shelf-safe milk can sit in your cupboard until you're ready to drink it. Then you take it out, you put it in the fridge so that it cools off. Most of us don't like warm milk. And then open it when we're ready, and we just drink it down just like normal. It tastes the same as our traditional milk. It's just stored differently. And it's actually the common mechanism of storage of milk in Italy, in I believe Germany, and in Chile. So that's how they store their milk. And then you just grab a carton out of the cupboard. So we can also use moist heat to sterilize. So when we use moist heat to sterilize, we actually have to get the temperature higher than just boiling. So we use, in an autoclave, what we do is we use, we increase the pressure. And when you increase the pressure in the system, it causes the temperature to rise. And this allows us to kill off microbes that normally would not be able to be killed off. This is what we do to sterilize all of our media. It's also what we do to get rid of the bacteria that are being grown in the lab. Oh, I didn't say this, I want to. So sterilization is usually about 15 minutes. And then this is at 121 degrees Celsius or 250 degrees Fahrenheit. When we are getting rid of bacteria, we tend to run the autoclave for 30 minutes, just to make sure that we're getting rid of everything. We can also use filtration. Filtration is not, so I should back up and say, okay, we're leaving the heat and now we're moving on to the next mechanism. This is a mechanical removal of microbes. Filtration is a very effective mechanism of removing microbes, especially if the microbes are in the air. So here you're seeing a filtration trap where fluids move through a filter and the larger microbes can't get through. And so what we have down here is a sterile liquid. Over here we have a filter that actually allows us to count the number of microbes. So on here we could actually take this filter, the bacteria are on it, and then we can put it into a media, and all the bacteria that are growing, we can identify them and allow us to know what in the solution was growing. We can also use filters that suck air in and get rid of the potential, dangerous bacteria in the air. HEPA filters are used that way. A lot of people use filters on their, their furnaces to get rid of any potential dust and material as well as bacteria. Radiation can be used to control microbial growth. So there are different types of radiation. The form of radiation the different forms, gamma radiation or irradiation and UV radiation are the three types that I typically talk about. Irradiation and gamma radiation really are about the same, whereas UV radiation is a little bit different. So UV radiation affects microbes, but it's not strong enough to go through glass. It's not strong enough to go through plastic. So if you're sitting inside a car and UV light is hitting the windows, it's not going to cause you to get burned or anything. But if your arm is sticking out and that UV radiation is hitting your skin, it can cause burns. Well, in microbes, what UV radiation does is it causes a thymine dimer to occur. Thymine dimers occur when you have two thymine in your DNA that are sitting next to each other on the same chain, and then they just attach to each other instead of attaching to their mate or their, what do I normally say, their complement. So thymine and adenine normally bind, but in this case, the thymines that are sitting next to each other bind together, and then the adenines can't bind. And this causes DNA not to be able to be replicated or transcribed properly. Gamma radiation uses ionizing radiation, X radiation and gamma radiation uses is considered ionizing radiation, where we get rid of electrons. We force electrons out. And this causes bacteria, so we're getting rid of electrons, we're causing free radicals to kill off the cells, basically. And this affects metabolism, it affects protein production, it affects the growth of the organism. And so irradiation is a form of gamma radiation where we shoot radiation into fruits and vegetables, and then that kills off bacteria that typically would cause food spoilage. And this allows us to keep our fruits for longer periods. So if you purchase fruits and vegetables from the grocery store, they typically last longer than if you purchase fruits and vegetables from a farmer's market. And the reason for that is the fruits and vegetables at a farmer's market are not irradiated, so they don't last as long. It's one of those beneficial things that allows us to keep our foods for a longer period of time. But again, you know, what's the side effects? We don't know. And so here's kind of joke, but it really deals with, you know, what's going to happen in the future. Your Honor, when my client agreed to be irradiated, he was assured that nothing could possibly go wrong, and there he is. And Mr. Potato Head with nose and eyes and everything all messed up. The thing about this is, you know, we don't know what is going to occur in the future. That being said, it's really beneficial when you're trying to purchase foods, you know, for an entire week instead of having to go to the grocery store every day and pick up the fruits or vegetables that you might need. So, I mean, there's benefits on both sides. We have to, you know, does the cost outweigh the benefit? That's really what we're looking at there. We can use preservation methods, stop spoilage from occurring. Typically, we're either going to use low temperature, so that's one of the most common mechanisms. Low temperature decreases microbial growth because it stops the microbes from undergoing metabolic activities. Think about when your body is cold, it doesn't work as effectively, which is why anytime you're in a race or if you're in any type of sport, you always have to stretch and warm up first so that you can work effectively. So if we either put our food in the refrigerator or into a freezer, it slows down or stops microbial growth. But that doesn't mean the microbes always die. Typically, if you take it out and let it sit on the counter, the microbes will start growing again. We can dry out microbes as well. So basically dehydrate them. Salting or adding sugar can actually decrease the amount of water, so it dehydrates them, and dehydration typically causes death of the microbes. So we've talked about these different mechanisms, and Now we're going to go into the chemical control of microbes. Hold on a second. I want to see how many slides so I can decide if I want to stop. All right, so I just checked. I only have 14 slides left. So this video will probably be about a 40-minute video, but you can always watch it in 2 parts. You don't have to watch it all at once. So we're going to go into chemical control of microorganisms. And so we didn't really start using chemicals until around the 1800s. Prior to that, you know, we always washed tables and stuff. I mean, people kept their houses clean, but they didn't use chemicals to sterilize environments because they didn't really know that there were microbes, and specifically pathogenic microbes, around. And because of this, we didn't really worry about that stuff. But now that we know, we're using chemicals depending on what environment we're in will determine the type of chemicals we use. So antiseptics are used to destroy pathogens or microbes on living tissue, whereas disinfectants are used to destroy pathogens or microbes on non-living surfaces. So here we have an example of, you know, different cleaning products. These cleaning products we'd use on surfaces. We would not use them on living tissues. So one of the things we're going to, or these are different factors that we consider when we're looking at what chemical to use. How susceptible would the microbe be to that chemical? What temperature and what pH does the chemical work best at, and can we use it in the environment that we're going to use it in? What concentration do we have to use that chemical at? Is the concentration so great that it would become toxic to the individual? What environment are we using the chemical in, and what effects will the chemical have on the environment? And then we have to look at the type of microbes, so endospores, and this kind of goes back to that microbial susceptibility. Endospore formers are not going to be as susceptible to certain chemicals as other microbes. So to be an effective disinfectant or antiseptic, you need to be able to slow the growth or kill microbes, but also be non-toxic. So you need to kill the microbes, but not hurt the animals, the humans, the plants, and the environment. Typically, you want to be dissolvable in water or alcohol and easy to apply. We like our chemicals to have a long shelf life, so we don't have to buy new chemicals every month. The more dilute we can use, the better, because we have less chemical per water. How fast does the chemical work? Does the chemical cause damage to the material that it's going to be on? Does the chemical have toxic smells? And how expensive is the chemical? One test that we typically use to evaluate disinfectants and antiseptics is the phenol coefficient test. So we compare things to a known phenol. And so an example of a phenol is Lysol. Lysol is a phenol. So we look at how well does phenol kill some microbe or some, you know, bacteria, and then we compare that to these other chemicals. And if they have a better ability to kill off the microbe, then they're considered good. If they can't kill the microbe off as effective, then they're not considered as good. Another mechanism that is used often especially in laboratories, is a disk diffusion test, where we look at a specific microbe if we're concerned about, say, an outbreak of some pathogen. We might look at how that pathogen grows in different environments. So here we just, there's some bacteria growing all over this plate. It's been basically spread completely to cover the entire plate. And then we put discs. These were sterile discs that we put different chemicals on. So mouthwash, iodine, alcohol, hand sanitizers. And what we do is we then allow these to sit in the incubator and then check that out later. And so what we can see is that bleach is very effective at controlling the growth of whatever this microbe is, compared to this kitchen cleanser. So if we were going to be cleaning our kitchen, we may want to use a 10% bleach solution versus the kitchen cleanser. If we're washing our hands, hand sanitizer and water aren't working very effective, we might want to use an alcohol-based solution. Or we might want to use iodine to get our hands more clean. We wouldn't use any of these really on our mouth. Maybe water, and mouthwash is actually better than water. So I talked about these already, but here's the list again. Toxicity of the chemical, how it works, so the environment it's working in, the compatibility with the material, cost, storage, and the environment. So alcohols are relatively inexpensive chemicals, and they work decently. They're very, very quick workers, but they evaporate just as quickly. So if you're using an alcohol to clean your counter, it's going to evaporate very rapidly. What alcohols do is they denature proteins. Aldehydes. So there are different types of aldehydes. Most of you have heard of formaldehyde. That's the chemical that is used to preserve tissues. So like hearts and brains and bodies. We use formaldehyde as our preservative, and then once it's preserved, we can then stick it into a different solution that is not as chemically intense, it doesn't smell as disgusting as formaldehyde, and it's not as likely to cause cancer as formaldehyde can. So aldehydes inactivate proteins and new nucleic acids. They can sterilize the environment. And they are used to typically in preservation processes, as well as, other things, but the main one that in biology we use formaldehyde for, or aldehydes, is for preserving specimens. It's not effective against all specimens though, so we can preserve these worms, and these worms, if we kill them, they will be in the solution, the formalin solution, but their eggs are still viable, which means the eggs can still hatch. The eggs don't die in the aldehydes. And so if you were dissecting these helminthic worms and you inhaled an egg, because these eggs are microscopic, you could actually get that worm in your system. It's kind of creepy. We'll talk more about that worm. The worm that you're looking at there is called Ascaris lumbricoides, and it causes the disease ascariasis. So biguanides, these are probably more commonly known to you than the aldehydes because you use them. They're found in things like mouthwash, in antiseptics, in lotions. They're found in soaps. One that we use in our lab was chlorhexidine, which is also used in hospitals. Oh, I'm sorry. This kills all vegetative bacteria, but does not kill endospores. It can get rid of certain viruses and fungi, but non-enveloped viruses are not killed off by these biguanides. Ethylene oxide, this is a gas, and it's a sterilant. So it sterilizes the environment, gets rid of endospores, viruses, everything. It's very useful at treating materials that are either heat sensitive or moisture sensitive. So it can sterilize metals, it can sterilize things like hospital items that we need to sterilize. The one thing that we have to, be careful about is ethylene oxide is considered a carcinogen. And so we would have to, anything that is going to be gassed by ethylene oxide would have to go into a special chamber. And then once it's closed, then we sterilize, and then we allow that chamber to vent so that we don't suck in that gas. Halogens. Halogens are oxidizing agents, so they oxidize proteins as well as cell structures like the plasma membrane. And so oxidation means that you're going to basically tear apart the plasma membrane or those proteins. The two most common halogens are chlorine and iodine. Oh, sorry. And you can see here, chlorine is used in the water to treat and get rid of any potential pathogens in water. Iodine is very commonly used in treatment of the skin, especially prior to surgical procedures. So chlorine destroys microbes in water, including viruses, and so it makes our water drinkable. It's treated. We don't have to worry about any disease. We're not going to get cholera from our drinking water, which is really a good thing because cholera will cause severe diarrhea, and severe diarrhea leads to dehydration, which would lead us to drinking more water, and if the cholera was coming from the water, then we would die. So very important that we have healthy hold on a second. Jeez, hold on. Sorry about that. I've got to finish this before that goes off because my computer's going to be restarting in about 10 minutes. Iodine in and of itself is rarely used straight up because iodine can cause burns on your skin. So instead, iodine is typically used with other organic molecules, and these organic molecules allow the iodine to be released slowly, and that reduces the likelihood of burns occurring. So iodine is bactericidal, fungicidal, and virucidal. So it gets rid of pretty much everything, but It does cause stains. It also can cause burns. And certain bacteria have evolved to be able to grow in the environment with iodine, which leads to a lot of hospital-associated infections. The last chemical we'll talk about are phenols. Phenols then are the one of the oldest chemicals that we have that helps clean environments. Typically, it has an unpleasant odor, so think about Lysol. When you spray it, it kind of has that weird odor. They have been making Lysol better, so I mean, 1980s Lysol is not the same as the Lysol of today, I will say that. It also can cause irritation to skin. It is effective at killing bacteria. It does not kill endospores though, so vegetative cells, yes, endospores, not always. It does get rid of a lot or inactivate a lot of viruses, but not all viruses are inactivated by phenols. And it can, the benefits though, because it's so effective at killing the majority of microbes out there, and it's relatively cheap, it works in the presence of most organic compounds. It's one of the most commonly used chemicals because of that. And I'm going to stop here. It's a 41-minute video. I do have other things I'll add on Thursday, but please come to the class with questions over both chapters 6 and 9, as well as chapter 4 or 5 if you have questions in that one, okay? So your exam, I know it's not coming up super quick, but it will be here before you realize it. All right, you have a wonderful night. Bye, and see you Tuesday.